In 2019, a groundbreaking publication by researchers at the Beijing Genomics Institute sent a ripple of profound silence through the global scientific community. They had meticulously sequenced the genomes of more than 140,000 Chinese individuals, benchmarking this vast data set against existing European genetic databases. The revelations were not merely surprising. They fundamentally challenged and reshaped established paradigms regarding human evolution, migration patterns, and the mechanisms driving genetic divergence. The results proved so contentious that the initial submission was rejected by 3 peer reviewers. Their objection was not rooted in any scientific flaw or methodological weakness. It was rooted in the sheer, weighty profundity of the paper's implications. To truly grasp the magnitude of these shocking discoveries, we must first understand the long-held assumptions scientists expected to validate. For decades, the mainstream understanding of human ancestry charted a straightforward course. Modern humans evolved in Africa approximately 300,000 years ago. Then, around 70,000 years ago, small founder groups embarked on a monumental migration, populating the globe. As migratory waves progressed, populations developed readily apparent physical distinctions, variations in skin color, eye shape, and hair texture. Yet, beneath these superficial differences, the prevailing scientific consensus maintained that, at a fundamental genetic level, all human populations remained largely uniform. Minor regional adaptations like lactose tolerance in Europeans or altitude adaptation in Tibetans were acknowledged. But the overarching genetic blueprint, it was widely believed, remained almost identical. It was against this backdrop of assumed uniformity that the BGI embarked on their ambitious project. Crucially, their initial motivation was never to stir controversy. Their objective was simply to comprehensively map genetic variations across China's enormous population of 1.4 billion people. The primary goals were practical: to advance healthcare, enable accurate disease prediction, and develop personalized medicine tailored to the unique genetic landscape of the Chinese populace. However, when researchers began comparing these Chinese genomes to established European databases, the numerical comparisons defied all expectations. The first major shockwave emanated from observations concerning genetic diversity. The range and variety of genetic differences within a population. Scientists had long anticipated that Asian populations would exhibit lower overall genetic diversity compared to African populations, a consequence of descending from smaller founder groups. This proved correct. However, what stunned researchers was the stark contrast in genetic diversity patterns between Chinese and European populations. European populations exhibited a relatively uniform level of genetic diversity across the continent. DNA from Sweden or Greece showed remarkably consistent levels of variation. In stark contrast, Chinese populations displayed a startlingly wide range of genetic diversity, varying dramatically depending on the specific region. Southern Chinese populations exhibited variation patterns entirely distinct from their northern counterparts. By certain metrics, the genetic distance between a Han Chinese individual from Beijing and one from Guangdong proved greater than the distance separating a Swede from a Greek. While this initial discovery of massive regional diversity was profoundly surprising, it was merely the prelude. As researchers delved deeper, analyzing specific gene variants, they uncovered thousands of genetic mutations showing dramatically different frequencies. These variants were either extremely common within Chinese populations, but rare in Europeans, or vice versa. Crucially, these weren't merely cosmetic distinctions. These were genes intricately involved in fundamental biological processes, impacting metabolism, immune function, brain development, and disease susceptibility. A prominent example that immediately captured attention was the gene variant EDARV370A. This specific mutation influences the development of hair, teeth, and sweat glands. Astonishingly, in Chinese populations, this variant is present in over 90% of individuals. In Europeans, its prevalence drops to less than 5%. This is no minor variation. It represents a fundamental genetic divergence. But this is precisely where the findings veered into highly controversial territory. The EDAR gene's influence extends beyond mere physical traits. It also plays a critical role in the branching patterns of neural connections during fetal development. This broader scope prompted deeply uncomfortable questions. If this impacts brain structure, what are the implications? The paper prudently refrained, but the question lingered. Then arrived the next discovery, a finding that truly shattered long-held assumptions. Scientists uncovered compelling evidence of widespread selective sweeps. A selective sweep occurs when a genetic mutation rapidly increases in frequency within a population because it confers a significant survival advantage. It is evolution operating in an accelerated, fast-forward mode. Such a mutation can propagate throughout a population in a mere few thousand years. European populations displayed evidence of selective sweeps primarily for genes associated with skin pigmentation and lactose tolerance. Readily understood adaptations to northern latitudes in dairy farming. However, Chinese populations exhibited selective sweeps in entirely distinct categories. These involve genes related to fat metabolism, neurotransmitter regulation, and even spatial reasoning and memory formation. Crucially, these selective sweeps occurred relatively recently, within the last 10,000 to 30,000 years. This time frame is profoundly significant. It falls well after the major migration out of Africa. This means these genetic changes were not inherited from a shared common ancestor. Instead, they evolved independently within Chinese populations after their divergence from European lineages. The implications were staggering. It suggested that populations had not merely diverged gradually through random genetic drift. They had been actively shaped by distinct evolutionary pressures, optimizing unique genetic profiles to thrive in respective environments. The discoveries pushed the boundaries of understanding even further when researchers found what they termed ghost DNA. This refers to genetic material that simply did not align with any currently recognized human lineage. It was not derived from the Homo sapiens who migrated out of Africa. It was not from Neanderthals, known to have interbred with European ancestors. Nor was it from Denisovans, who shared genetic material with some ancient Asian populations. It was something entirely distinct. While European populations typically carry an average of about 2% Neanderthal DNA, the Beijing study unearthed segments within Chinese genomes that failed to match any known archaic human species. Although the percentages were small, their consistent presence was undeniable. From where did this mysterious DNA originate? One compelling hypothesis posits the existence of another archaic human species in Asia, one that remains undiscovered by modern science. Under this scenario, ancient Chinese populations would have interbred with this unknown group. An alternative hypothesis suggests this ghost DNA might stem from convergent evolution, similar genetic mutations arising independently to address analogous environmental challenges. However, this explanation struggles to fully account for the highly specific genetic patterns observed. Regardless of its precise origin, the undeniable fact remains: Chinese populations possess unique genetic material not found in Europeans, pointing to a much more complex ancient history. It is at this juncture that the controversy truly erupted. Recall that the impetus for this research was fundamentally medical. The goal was to enhance healthcare by understanding genetic differences influencing disease risk. Within the realm of practical medicine, the findings became impossible to overlook. Chinese populations exhibited significantly different genetic risk profiles for virtually every major disease category when compared to Europeans. This encompassed cardiovascular disease, diabetes, Alzheimer's, and various cancers. The genetic variants known to elevate disease risk in European populations frequently demonstrated entirely different effects in Chinese populations, or were absent altogether. Consider type 2 diabetes. In European populations, The most potent genetic risk factors are variants within genes such as TCF7L2. These dramatically heighten diabetes risk among Europeans. However, in Chinese populations, these very same variants displayed significantly weaker associations. Instead, the risk of type 2 diabetes in Chinese individuals was largely driven by an entirely different set of gene variants, which held minimal relevance in Europe. This finding carries massive, transformative implications for the entire field of medicine. For decades, the vast majority of drug trials and medical research have been predominantly conducted on populations of European descent. Drugs proven effective in Europeans were broadly assumed to be universally effective. This study proved that assumption dangerously false. If the underlying genetic architecture of disease is fundamentally divergent between populations, then drugs optimized for Europeans might not function the same way in others. This is precisely what researchers began to uncover. Certain medications for high blood pressure demonstrated reduced efficacy in Chinese patients. Chemotherapy drugs yielded different response rates. Even common pain medications were metabolized differently. The global medical community was compelled to confront an undeniable truth. Population-level genetic differences are real, significant, and clinically relevant. One of the most fascinating follow-up discoveries emerged from investigating diet-related genes. Researchers discovered that Chinese populations exhibited distinct genetic adaptations to high-starch diets, contrasting sharply with European adaptations to dairy. The AMY1 gene, responsible for an enzyme that breaks down starches, displayed significantly more copy number variations in Chinese populations. More copies meant greater enzyme production and more efficient starch digestion. This adaptation makes perfect evolutionary sense. Rice cultivation in China dates back over 10,000 years. A consistently high-starch diet exerted strong evolutionary pressure, favoring individuals who could efficiently metabolize carbohydrates. Meanwhile, Europeans developed lactase persistence to digest milk sugars in colder northern climates. These distinct dietary adaptations created unique metabolic baselines that continue to persist in modern populations, directly impacting insulin response and cardiovascular health. Adding another layer of complexity is the connection to the human microbiome. Gut bacteria are influenced by both dietary habits and underlying genetics. Chinese populations exhibit different gut bacterial compositions compared to European populations. These differences are partly driven by the very genetic variants affecting digestion and metabolism. This establishes a dynamic feedback loop. Genes shape the microbiome, and the microbiome influences health outcomes. The entire intricate system is significantly population-specific. We also see this localized fine-tuning in altitude adaptations. While Tibetans possess an extreme adaptation through Denisovan DNA to survive high elevations, Han Chinese populations living at sea level also carry different variants in oxygen metabolism genes compared to Europeans. These variants appear to optimize oxygen usage in subtly different ways. Genetic differences are not solely about adapting to dramatic environments. They reflect a sophisticated fine-tuning of metabolism, precisely adapted to specific conditions endured over thousands of years. Publishing these groundbreaking medical findings, however, inadvertently opened a Pandora's box. Once science formally acknowledged that human populations differ genetically in ways impacting health, the unavoidable question arose: what else might be different? The researchers were acutely aware of the political and ethical minefield they were navigating. The paper meticulously avoided discussion of cognitive traits, or anything that could be interpreted as ranking populations. Yet the raw data presented undeniable patterns: gene variants affecting neurotransmitter function, and variations impacting brain structure development. As mentioned, 3 peer reviewers initially rejected the paper. Their concerns centered on the potential for these findings to be misinterpreted or deliberately misused to fuel racist narratives. The research team pushed back forcefully. They contended that suppressing scientifically accurate data for political reasons was unethical. If genuine genetic differences exist and impact medical outcomes, healthcare professionals absolutely need to be aware of them. Ignoring these realities would only lead to suboptimal healthcare. The paper was eventually published with extensive caveats. The authors rigorously emphasized that genetic differences between individuals within a single population are vastly greater than the average differences between populations. Two random Chinese individuals differ genetically from each other far more than the average Chinese person differs from a European. The study demonstrated that the pattern of genetic variation differs, reflecting distinct evolutionary histories. Crucially, this does not establish a hierarchy. It illuminates diversity. Yet the unsettling questions from neuroscience research remain. Genes like ASPM and MCPH1, crucial for brain size regulation, exhibit distinct variant frequencies between these populations. Do these genetic distinctions imply anything meaningful about complex human cognition? The scientific consensus is that we simply do not yet know. Most cognitive abilities are influenced by thousands of genes alongside powerful environmental and cultural factors. Attempting to draw definitive conclusions from a handful of variants is scientifically premature. But we do know brain development is influenced by genetics. If populations exhibit significant differences in these foundational systems, there is a theoretical possibility that population-level patterns possess underlying genetic components. Most researchers refuse to touch this topic due to immense political risk. However, the BGI study opened a door that cannot be closed. We now possess concrete evidence of distinct evolutionary trajectories. The paramount question becomes, what will we do with this knowledge? The optimistic perspective points to a revolution in personalized medicine, with treatments custom designed for specific genetic ancestries. Pharmaceutical companies are now routinely conducting separate drug trials in diverse populations. But there lurks a darker possibility. Could this knowledge be weaponized to justify discrimination? The science itself is inherently neutral. The identified genetic differences are demonstrably real and objectively measurable. Real does not imply superiority. It simply denotes biological variation. The results shocked everyone. Now, it is up to all of us to decide how this profound understanding of human diversity will shape our future.